Hi guys, so today we are talking about passive transport. Passive transport ties right in with this objective right here that we will be looking at the method of transport in cells. So diffusion and osmosis are both types of passive transport. Passive transport is the movement of molecules across a cell membrane without energy. So if you think of when you're sitting at your house and you're just watching TV or reading a book, you are being passive. You're not exerting any energy. And that's how we can remember passive transport, no energy. Visually, here is what passive transport looks like. So we have it in two different forms. This would be diffusion here. Molecules just moving across the membrane. We see no energy input from the cell. We're just moving across the membrane. And then we have something called facilitated diffusion. This is the same thing as diffusion, except we're using a protein to cross the membrane. Diffusion is the movement of molecules in a fluid or a gaseous state from a region of high concentration to a region of low concentration. So we're moving down the concentration gradient. We want to be in equilibrium where the amount of molecules both inside the cell and outside of the cell are the same. The concentration gradient is the difference in a concentration from a substance in one location to another. Osmosis is a special type of diffusion that is named to how water moves across a membrane. The movement of water molecules across a semi-permeable membrane from high concentration to low concentration is osmosis. Water cannot move across the membrane without help, though. Water does need a specialized protein to help it move across the membrane through osmosis. We have three main types of osmosis. We can be an isotonic, where the solution has the same amount of particles both inside and outside of the cell. So as one moves in, another one moves out. So that's isotonic, meaning the same. We have hypertonic. This is a solution that has a higher concentration of dissolved particles outside of the cell than inside of the cell, and therefore water leaves. This is messy. So water leaves our cell. Leaves our cell in hypertonic solutions. In hypotonic solutions, the solution has a lower concentration than that of the cell. So we have water is gained by the cell. In our hypotonic solution, the cell gets very large, like a balloon. It swells up because water is being gained by the cell, but in a hypertonic solution, the cell shrivels up because the water is leaving the cell. Let's erase that. This is an isotonic solution here. We use red blood cells as the best way to show this because red blood cells are very simple. We want them to be in an isotonic solution so they can carry enough oxygen in the middle of this donut to the extremities of the cell, of the body, and then return carbon dioxide back to the lungs so we can rid it. In a hypertonic solution, those cells are shriveling because they are losing too much water. This can lead to diseases such as sickle cell anemia, blood clots, anything like that. And then in a hypotonic solution, the cell is gaining too much water that it will inevitably burst. Just again, hypotonic, not enough outside, too much inside. Isotonic is the same. Hypertonic, too much outside, not enough inside. Molecules can also diffuse through transport proteins. This is called facilitated diffusion. This is the diffusion of molecules across a membrane through transport proteins. Transport proteins make it easier for molecules to enter and exit the cell. Molecules move down their concentration gradient still through diffusion. It's still a passive transport, so there is no energy input from the cell. And we are going to stop here. We will cover active transport in our next video.